Hello viewers, in the last video we studied about the cellular responses to stress and injurious stimuli. In this video we will be studying about the mechanisms of cell injury. So what are the intracellular targets of cell injury? What are the principal intracellular targets of cell injury? Mitochondria, cell membrane, machinery of protein synthesis and secretion, and DNA. So coming on to the first, mitochondrial damage. Mitochondria play important role in cell injury and cell death. They are susceptible to almost all types of cell injury. Why? Because they are providing the life-sustaining energy in the form of ATP. So what are the various types of injurious stimuli that can lead to mitochondrial damage? Ischemia, there is decreased oxygen, then chemical injury, chemical agents, reactive oxygen species, and increase in cytosolic calcium levels. So they can all cause, and various other types of injurious stimuli, they can cause mitochondrial damage. So mitochondria, they are producing ATP. So what will be the effect of mitochondrial damage, or what will be the consequences of mitochondrial damage? There will be ATP depletion. There will be increase in formation of reactive oxygen species. Reactive oxygen species. And third, there will be channel formation by pro-apoptotic back and backs proteins that will lead to release of mitochondrial proteins into the cytoplasm. So coming on to this ATP depletion, how ATP is produced? So the ATP is produced by two pathways. The major pathway is by oxidative phosphorylation. And other is the anaerobic glycolysis. So this is the major pathway, so it requires oxygen. Now what is the role of, why ATP is so important? ATP is required for several synthetic and degradative functions in the cell. What are these? It is required for protein synthesis. It is required for membrane transport. For acylation deacylation reaction. which is necessary for phospholipid turnover. Phospholipid turnover. And then lipogenesis. So what happens when the ATP levels fall below 5 to 10% of the normal values? So when they fall below 5 to 10% of the normal value, then this leads to a variety of deleterious effects on the cellular functions. So let us study about them one by one. First is, there is decreased activity of sodium potassium pump. So this pump requires ATP for its normal function. So when it is functioning normally, it is transporting 3 sodium ions out of the cell in exchange of 2 potassium ions. So 2 potassium ions are coming inside the cell. So when this activity is decreased due to depletion in ATP, there will be net gain of sodium inside the cell, and with it will come water. So there will be net gain of solute as well as water. So there will be net gain of sodium and water. So what will be the effect on the cell? There will be cellular swelling. This will lead to cellular swelling and dilation of endoplasmic reticulum. So next effect of depletion in ATP is cellular energy metabolism is altered. Cellular energy metabolism is altered. What does it mean? It means that normally energy was being produced by oxidative phosphorylation. So when there is depletion of oxygen, cell has to resort to some other method for production of ATP. What is the other method? Anaerobic glycolysis. So it utilizes glucose as a substrate and leads to generation of ATP, but the amount of ATP generated is very less as compared to that generated by oxidative phosphorylation. So with it there is production of lactic acid and inorganic phosphates. So this lactic acid, it leads to decrease in the pH of the cell. pH of the cell gets decreased, and due to this decreased pH, there is decreased activity of various cellular enzymes. Third is decreased protein synthesis. As ATP is required for protein synthesis, so in the absence or depletion of ATP, there will be decreased protein synthesis. The protein synthetic apparatus will be disrupted. What does it mean? There will be detachment of ribosomes from the rough endoplasmic reticulum, and also there will be dissociation of polysomes. Dissociation of polysomes. And there will be increase in misfolded proteins. Misfolded proteins. So when the proteins or the polypeptide chains they are produced, they have to be properly folded for the proper function. So this is done by foldases, enzyme foldases. So these foldases also require ATP for proper function. So when ATP is not there, there will be increase in the misfolded proteins, and this will lead to ER stress, which is a trigger for apoptosis, Fourth one is irreversible damage to mitochondrial membrane and lysosomal membrane, leading to necrosis. Also, when mitochondrial damage occurs, there is formation of mitochondrial permeability transition pore. Mitochondrial permeability transition pore. When this pore is formed, there is loss of the membrane potential, and this will further lead to decrease in the ATP synthesis. And through this pore, various mitochondrial proteins, they leak out into the cell. So coming on to the second this consequence of mitochondrial damage, which is reactive oxygen species. So coming on to the next consequence of mitochondrial damage, reactive oxygen species. So what are reactive oxygen species? These are oxygen derived free radicals. What are free radicals? Free radicals are chemical species that have an unpaired electron in the outer orbit. So having unpaired electron in the outer orbit, this is an unstable configuration. So these free radicals, they are highly reactive species. They react with inorganic and organic molecules, and they convert the molecule into a free radical. They attack the molecule, and the molecule is itself converted into a free radical. So they propagate a chain of damage. So what are the oxygen derived free radicals? These are superoxide anion, hydrogen peroxide, and hydroxyl free radical. These are the main oxygen derived free radicals. So free radicals, they are also a part of normal cellular respiration. They are being normally produced during mitochondrial respiration. But the cell has scavenging mechanisms to clear out these oxygen derived free radicals. So whenever there is excess production of oxygen derived free radicals, or the scavenging system, it gets inadequate, so there will be oxidative stress. So during cellular respiration, what is happening? During cellular respiration, there is reduction of oxygen by transfer of 4 electron to H2 molecule to generate 2 molecules of water. When there is incomplete transfer of electrons in superoxide, when there is transfer of only 1 electron, this will lead to formation of superoxide anion. When there is transfer of 2 electrons, there will be formation of hydrogen peroxide, and hydroxyl free radical is formed when there is transfer of 3 electrons. So this hydroxyl free radical is highly reactive. This is highly reactive. So what are the effects of reactive oxygen species on various cellular components? So these reactive oxygen species, they act by various mechanisms. They cause lipid peroxidation of membranes. Lipid peroxidation of membranes. They attack the double bonds in the unsaturated fatty acids of the lipids in the membranes, and they form peroxides. And these peroxides, then they react with the other molecules and they propagate a chain of damage, which is called as propagation. Thus they cause damage to the membranes. Second is oxidative modification of proteins. These reactive oxygen species, they cause oxidation of the amino acid side chains, the protein backbone, and they form covalent linkages between proteins. So they form disulfide bonds between proteins. So what will happen due to these oxidative modifications? So there will be, in case of enzymes, the active site of the enzyme, it gets modified or it gets lost. So enzymes, the active site of the enzymes, it gets modified. So the enzyme action will not be there. And also in structural proteins, there will be some changes in the conformation of the protein. The structural proteins. And this modification will lead to a burden on the ubiquitin proteasomal pathway, which causes degradation of the misfolded proteins. So this will further lead to ER stress. Next is lesion in DNA. Lesion in DNA. So these reactive oxygen species, they cause single-stranded or double-stranded breaks in the DNA. Single or double-stranded breaks in the DNA. And they form cross linkages leading to adduct formation. So these are the mechanisms by which reactive oxygen species, they cause cell injury or they cause cellular damage. So now all these mechanisms, the injurious stimuli, they are triggering multiple interconnecting pathways simultaneously, and these are all interrelated. So one pathway, due to one pathway, the action of the or the damage caused by other pathway is enhanced. So all these are interrelated. So now we will, with the help of a diagram, we will see the role of mitochondria in cell injury and cell death. This is mitochondria. Now the injurious stimuli in the form of ischemia, in the form of toxins, radiations, they cause mitochondrial damage, which will lead to decrease in the ATP decrease ATP and increase in the reactive oxygen species. So both of these will lead to multiple cellular abnormalities, which leads to death by necrosis. Now here, so basically what was happening here? There was depletion of oxygen. Now coming on to this, whenever there is DNA or protein damage, or there is a decrease in the survival signals, there will be increase in pro-apoptotic molecules and decrease in the anti-apoptotic molecules, which will lead to release of pro-apoptotic molecules into the cytoplasm, molecules in cytoplasm, and this will trigger apoptosis. So mitochondria plays important role in cell injury as well as in both the major pathways leading to cell death. Next mechanism, cell membrane damage. So cell membrane damage is seen in almost all types of injurious stimuli. So it is seen in ischemic injury due to exposure to viral proteins, bacterial toxins, lytic complement components, physical agents, physical and chemical agents. So they cause direct cell injury, physical and chemical agents. So there can be early loss of selective membrane permeability leading to overt membrane damage, and this is seen in all types of cell injury leading to necrosis. In cell injury leading to apoptosis, in apoptosis cell membrane damage is not a feature. Cell membrane damage is not seen in apoptosis, but it is a feature in all injurious stimuli which lead to necrosis, cell death by necrosis. So what is happening in ischemic injury? So there is depletion of ATP and there is calcium mediated activation of phospholipases. So we have understood why this ATP depletion occurs in ischemic injury. We have studied about it in the mitochondrial damage. So why this calcium mediated phospholipase activation occurs? Now calcium, there is a calcium homeostasis in which the intracellular calcium level is maintained at low concentration as compared to extracellular levels. Intracellularly the levels are less than 0.1 micromoles and extracellularly they are 1.3 millimoles. So whenever there is ischemic injury or injury due to some toxins, there is disturbance in this calcium homeostasis. So what will happen? The extracellular calcium will move inside the cell and the intracellular calcium or the cytosolic calcium, it is sequestered inside endoplasmic reticulum and mitochondria. So there will be release of sequestered calcium from these sites into the cytoplasm. So let us understand it with the help of a diagram. So this is a cell. And calcium is sequestered inside mitochondria and endoplasmic reticulum. So due to injurious stimuli, first there is release of calcium from these two sites, and later on there is calcium entry inside the cell from the extracellular source. So this calcium leads to activation of cellular enzymes. Increased calcium leads to activation of cellular enzymes. What are these enzymes? These are phospholipase. So it will lead to breakdown of phospholipids which are constituent of the cell membrane. So it will lead to phospholipid breakdown. Protease, they will lead to cytoskeletal breakdown. The cytoskeletal filaments, they will be degraded. Endonuclease, so they cause nuclear damage. ATPase will lead to depletion of ATP. Now this increase in cytosolic calcium, it has one more effect. It leads to formation of mitochondrial permeability transition pore in the mitochondria. Leads to mitochondrial damage. Mitochondrial permeability transition pore formation occurs, and when this pore is formed, there will be depletion of ATP. There will be depletion of ATP, decreased ATP, and also there will be release of pro-apoptotic molecules into the cytoplasm, and this will further lead to depletion of ATP. So there will be decreased ATP, there will be nuclear damage, and both these lead to cell membrane damage. So this is the role of increased cytosolic calcium in cell membrane damage. Now let us study the various biochemical mechanisms that lead to cell membrane damage. So these mechanisms are first is reactive oxygen species. So reactive oxygen species damage the cell membrane by formation of lipid peroxidation. Second is there is decreased synthesis of phospholipids. Why phospholipid synthesis is decreased? Because there is decrease in ATP. ATP is required for various synthetic activities, for the acylation reaction which is required for phospholipid synthesis. So due to injury, when ATP depletion occurs, there is decrease in the synthesis of phospholipids. Increased phospholipid breakdown. Why this increase in phospholipid breakdown? Due to calcium mediated activation of phospholipases. So these phospholipases, they break down phospholipid to form lipid breakdown products. And what are these lipid breakdown products? These are unesterified free fatty acids, acyl carnitine, and lysophospholipids. Lysophospholipids. These have a detergent action on the cell membrane. These have a detergent action on cell membranes. Also, they can get inserted in place of phospholipids, and they alter the osmotic permeability or the membrane permeability of the membranes. So this leads to cell damage. Next is the cytoskeletal abnormalities. Cytoskeletal abnormalities. So these cytoskeletal filaments, they act as tethers. They bind the plasma membrane to the interior of the cells. So when there is calcium mediated activation of proteases, so they lead to breakdown of these tethers. So the cytoskeletal filaments get broken down, and this leads to, and if already there is cellular swelling, this will lead to rupture of the cell. This can rupture the cell. So these are the 4 biochemical mechanisms by which cell membrane damage occurs. Now what are the various effects of this cell membrane damage? Because cell membrane is not only the plasma membrane, it is covering all the organelles of the cell. So organellar membrane will also be damaged when cell membrane damage occurs. So what are the effects of cell membrane damage on the cell? So what will happen when there is mitochondrial membrane damage? So mitochondrial membrane damage will lead to formation of mitochondrial permeability, transition pore, which will lead to depletion of ATP and release of mitochondrial proteins into the cell. Plasma membrane damage. What will happen when the plasma membrane damage has occurred? So it will lead to the loss of the osmotic permeability. There will be influx of ions, fluids, and there will be loss of cellular contents. There will be loss of metabolic substrates. Now what will happen when lysosomal membrane get ruptured? Lysosomal membrane damage. So what is stored in the lysosomes? Lysosome has acid hydrolases. So these acid hydrolases, they come out. Acid hydrolases, they are released inside the cell and they get activated in the acidic environment of the cell. They get activated in the acidic environment. So how this acidic environment of the cell occurs? It is because of the lactic acid formation. The pH of the cell has decreased. So when these acid hydrolases, they come out, they get activated. What are these acid hydrolases? These are RNase, DNase, protease, phosphatase, and glucosidase. So RNAs will degrade RNA, DNAs will degrade DNA, protease will degrade proteins, phosphatase will degrade phosphoproteins, and glucosidase will degrade glycogen. And ultimately the cell will pushed towards necrosis. So this was about cell membrane damage. So let us understand the mechanism of cell membrane damage with the help of a diagram. So this is a cell. And this is the mitochondria here. So when mitochondrial damage occurs, there is depletion of ATP. Suppose it is due to ischemic injury. So depletion of ATP, and there is formation of reactive oxygen species. There will be depletion of ATP, which will lead to decrease in phospholipid synthesis, decreased phospholipid synthesis. And what will this cause? This will lead to lipid peroxidation of membranes. This will lead to lipid peroxidation of membranes. Also, there is increased cytosolic calcium level due to injury. So they will activate enzymes. Which enzymes? Phospholipase activation. Phospholipase activation. Protease activation. So phospholipase activation will lead to further depletion of phospholipid, and it will lead to formation of lipid breakdown products. So there will be phospholipid loss, and these three, they will lead to cell membrane damage. And this protease, this will lead to cytoskeletal abnormalities. It will also lead to cell membrane damage as well as breakdown of the cytoskeleton. So with this diagram, we have understood the mechanism of cell membrane damage. Now next, coming on to machinery of protein synthesis and secretion. This we have discussed already when we were discussing the mitochondrial damage. Next we come on to the DNA damage. Now next is DNA damage. So DNA can be damaged because of chemotherapeutic agents, due to radiations, reactive oxygen species, and also in aging DNA damage occurs. So what happens when DNA gets damaged? There is activation of p53 dependent pathway. p53 dependent pathway. So this p53, it causes cell cycle arrest in the G1 phase. Why cell cycle arrest occurs? In order to repair DNA. Now if it is successful, if DNA repair happens successfully, then the cell will survive. Now when DNA repair fails, so p53, it triggers cell death by apoptosis, by the mitochondrial pathway of apoptosis. So here the cell chooses to die instead of surviving with defective or damaged DNA. Now what happens when there are p53 mutations which interfere with cell cycle arrest and the induction of apoptosis? So obviously there will be DNA damage in this case. So in p53 mutations we see numerous cancers. So it will lead to neoplastic transformation. Numerous cancers are seen in p53 mutations. So these were the mechanisms by which cell injury occurs. In the next video we will be studying about necrosis. I hope you liked the video. Thanks for watching.